Right, protein synthesis. Okay, this presentation looks at the first few slides in your protein synthesis notes, and the first slide here deals with defining what the term gene means. Okay, so the first line here says a gene is a length of DNA or a sequence of nucleotides that codes for one polypeptide. If I was to look at the idea of a gene on this diagram here, what we see is that a gene is a length of DNA or a sequence of nucleotides, and this is best shown actually when the cell is just about to divide, as we know from the work you have done with Mrs. Cox. Cells just prior to dividing, this is eukaryotic cells, just prior to dividing, the DNA or chromatin as it's known, what it does is it condenses and forms up into these visible structures which we know as chromosomes. And chromosome, here is a chromosome here, with its two constituent chromatids. Okay? What a gene is, with respect to a chromatid or a chromosome, is it is a sequence of nucleotides or a length of DNA, okay, which exists at a particular position on a particular chromosome. Okay, and the term that we use for this position is the term locus, this term here. So a gene occupies a specific site on a chromosome which is termed a locus. Okay, so this could be the locus for a particular gene here. Okay, so this could be, for example, the locus for the gene that encodes for the protein insulin, for argument's sake. Okay, so that's where we find genes and what genes do, i.e. genes are a length of nucleotides which encodes for a particular polypeptide. Okay, now going back to your understanding of what genes were when you studied GCSE, you probably learnt a term along the lines of genes and the nucleus controls the cells. So how do they exert that control? Let's look at the next diagram here. The answer can be summarised in what is sometimes known as the central dogma of molecular biology, which goes some way to try to explain how DNA exerts control over cells and therefore exerts control over characteristics of individuals. If I look at this diagram here, this summarises this central dogma of molecular biology, which deals with DNA. Okay, DNA here can replicate, okay, and that allows copies of DNA to be put into new cells, which means that all the cells of an individual contain an exact copy of all of the DNA, or the genome as it's sometimes known, of an individual. Okay, so we've got copies of the DNA in all of the cells. Okay, that exerts influence on the cell, okay, by a series of information, a flow of information, if you like, okay, which goes from DNA, which in eukaryotes is found in the nucleus, okay, it is then transcribed, this word transcription, into a messenger molecule which is known as messenger RNA. Okay, that diffuses out into the cytoplasm of the cell, where it instructs the cytoplasm of the cell via ribosomes to make polypeptides, which, as we know, fold up to form particular proteins. And it's these proteins that do a whole series of functions, or perform a whole series of functions within the cell. The obvious example, the example we know the most about from our F212 course, was enzymes. We know that enzymes control chemical reactions and therefore the metabolism of a cell. Okay, so the DNA determines what proteins are expressed, and it's those proteins which determine what metabolism goes on inside the cell, and that metabolism determines its characteristic or phenotype. It's not just enzymes, things like carrier proteins are also very important in determining what goes on inside a cell. Moving on to the next slide, explain the meaning of the term genetic code. Okay, so there are various features that the code has which constitutes a gene. Okay, and we call that the genetic code. Okay, i.e. how the genetic code works and the features of this genetic code. Okay, first thing to appreciate, it's a linear code. Okay, so it works out on a sequence of bases, okay, that form part of the DNA. We can remember from AS that there are 4 bases that we encounter on DNA: adenine, cytosine, thymine, and guanine. And it's the sequence that these bases occur, in which these occur, which determine the genetic code. So it's a linear sequence of bases, which is eventually transcribed and translated, 2 words we'll come back to in the next slide, into a linear sequence of amino acids. Okay, and you remember from the work on proteins that you remember the primary, secondary, tertiary and quaternary structures of proteins. You'll remember that the key thing in a structure of a protein is its primary structure, which is its sequence of amino acids, and particularly those sequence of R groups, which determine the way that those amino acids, or those domains of amino acids in a polypeptide, fold up to form secondary structures and consequently tertiary structures. And if it's a complex protein with a quaternary structure, i.e. multiple polypeptide chains, they will bind around each other and form around each other, depending ultimately on the primary sequence of amino acids. So what we're saying here is that primary sequence of amino acids is dependent on the sequence of nucleotides that occur on the DNA. There are various features of this linear code you should be aware of. First of all, the idea it works in triplets. Okay, there are obviously only 4 bases. Okay, but we have to have a system which encodes for 20 different amino acids. Okay, so it actually works in triplets, and that would give us, this should say 4 to the power of 3 rather than 43, but anyway, a 4-letter language can give us 64 possible 3-letter words or triplet codes. Okay, but as there are only 20 amino acids and 64 possible codes, that means, for the mathematicians amongst you, there are 44 potentially redundant triplets. Okay, but it doesn't work like that, in that what we find is that each amino acid has more than one triplet code that codes for it, and that principle is known as the degenerate code. Okay, they're not actually redundant, because what we find is that there are multiple codes for each amino acid, and that's known as a degenerate code. We'll look at the importance, or one of the significances of this degeneracy when we come to look at mutations later on. One of the other features about it is it's widespread, almost universal, i.e. almost the same in all living organisms. Okay, but there are some living organisms that have slightly different genetic codes, some archaebacteria, and some of the coding that actually goes on in organelles of eukaryotes. So some mitochondrial DNA has a slightly different coding system than most. But most organisms have exactly the same genetic code, i.e. the same 3 bases in DNA leads to the same amino acid being deposited in the sequence of in a polypeptide. This, as we'll come across later in the term, is also forms the basis from which genetic engineering can occur, i.e. we can put a gene from one organism into another, and it's transcribed and translated in exactly the same way to make the same protein, which allows us to make things like insulin within bacterial cells and things like that. Okay, so a few diagrams here just to illustrate some of the things I've been talking about. First of all here, the idea of it being a linear code. It's a linear sequence of bases, here, which results in a linear sequence of amino acids here. Okay, so a linear sequence here leads to a linear sequence of amino acids here. What this diagram doesn't show is the intermediate messenger RNA molecule. Okay, it also says that the code forms in triplets. Okay, this 3 is red, and therefore this 3 is red, and therefore this 3 is red, and these triplets all result in the coding of a particular amino acid. Okay. One of the other features, which is I haven't mentioned in my slide, it's also described as non-overlapping, i.e. we read this 3 and then this 3 and then this 3, rather than another way of, it might have been organised but it's not, is it could have been this 3 followed by this 3 followed by this 3, okay? But it doesn't work like that, and that's described as being non-overlapping. Okay? If I actually start to look at the actual genetic code itself, okay, we actually know, you don't have to know any of these, okay, but there's interesting features to look at this. This shows you the actual codes for all of the amino acids, okay. This isn't actually the DNA codes, but this is actually the codes on the messenger RNA and which triplets are translated to which amino acids. So these things here are all the different amino acids, and they're three letters which denote, or symbols if you like, for each of the amino acids: phenylalanine, leucine, serine, proline, cysteine. Cysteine is that one which has got the sulfhydryl group we mentioned back in F212, and so on. And these, what you'll notice, the idea of degenerate code, is that you'll notice, for example, that leucine, okay, this amino acid here, has 1, 2, 3, 4 triplet codes. Okay, so these are RNA triplets, okay, on the messenger RNA, which leads to the translation in the polypeptide chain of the amino acid leucine. Okay, one of the things we'll look at a little bit later when we look at mutations is that you'll notice that all the triplet codes for leucine are all quite similar. Okay, so they all have for example, the first 2 bases there, and they also have the same second base there. So it's only in the third base that there's any difference in these triplet codes. Okay, one of the significances for that is if mistakes occur, it's very possible that a mistake might result in the same amino acid being put into the polypeptide. Okay, and therefore this code is said to be fault tolerant. So if CUU was accidentally transcribed into CUC, it wouldn't actually have any impact on which amino acid was placed into our polypeptide. Okay, so that's the actual genetic code. Again, you don't have to know any of these. Okay, there are other things which are quite useful to know about. You'll notice that there are one of these things, 3 of these things which say stop. Okay, there's a stop codons, okay, which actually signal at the end of a piece of mRNA, that at the end of a gene on a piece of mRNA, that actually the process of translation stops, it's interrupted, and the protein is actually completed by that stage. Okay, other ones, you'll notice that methionine down here only has 1 triplet code, or codon, as we'll talk about codons later on, only has 1 of these. All proteins start with this triplet code AUG, which leads to methionine being the first amino acid that is that's placed into each polypeptide. Okay, so quite a lot of proteins start with methionine. However, some proteins have the methionine chopped off in the Golgi body after the protein has been synthesized. Okay, but we'll look at that in a little bit more detail in a minute. Okay, this next slide looks at if you like, gives us an overview and introduces us to 2 key words. And the things I want you to take out of this slide are the keyword transcription and the keyword translation. Transcription is very simply the DNA to RNA, particularly messenger RNA, okay, and that happens in the nucleus. Okay, so the key idea to take out of that is this happens in the nucleus. Transcription in the nucleus, and that is from DNA is transcribed, okay, and DNA is used as a template to construct a molecule called messenger RNA, and that happens in the nucleus. Key idea. The next key idea I want you to take out of this slide is the idea that translation, this word here, translation, and that occurs in cytoplasm, particularly on ribosomes, which are often attached to endoplasmic reticulum, and that's called rough ER, when ribosomes are attached to endoplasmic reticulum. But that's when the messenger RNA, which we made in transcription, is read and results in a polypeptide. Okay, mRNA to polypeptide, and that is translation. As opposed to this, which here is transcription. Okay, so those are the two things I want you to take out of this slide. If I just show you that in a diagram I've got down here, which shows you what's going on. Okay, here we've got the nucleus of the cell. Okay, and this is where transcription happens, where we take DNA, which is in the nucleus, and we read that, okay, by transcription to make a molecule of messenger RNA. Okay, so that's transcription that occurs inside the nucleus. The messenger RNA, which is the product of transcription, then diffuses out of the cell through the nuclear pore. Okay, and here's the mRNA here. Okay, the mRNA attaches to a ribosome, which is this shaded structure here, and on that messenger RNA we'll look at in a little bit more detail, but transfer RNA molecules bring amino acids in the correct sequence according to the sequences of bases on the messenger RNA to make a sequence of amino acids, which are these little beads that represent amino acids, and this is a growing chain of amino acids to make a polypeptide. This is happening in the cytoplasm on the ribosome, and it's known as translation. Okay, so those are the key words I want you to take out of that slide. Okay, moving on to the... Right, this deals with the nuts and bolts of what's going on, some of which we studied at AS in the lower six. Okay, so I've got some diagrams here to demonstrate this, and also a few video clips for you to look at, because it is better visualized in sort of cartoon form. So, going back to my transcription to translation, okay, the first 1, 2, 3, 4 bullet points there describe the detail of what's going on in transcription. So the first thing is that, let's find a diagram I've got here. There's a diagram here. So if you look at you've got your hard copy of these notes somewhere, a hard copy of this slide up here. Got a hard copy of that slide up here. Here's a diagram which will hopefully illustrate that process of transcription, the first 4 bullet points on your notes. So what happens is that an enzyme called RNA polymerase, not to be confused with DNA polymerase, which you studied at AS, which is the enzyme which catalyzes the replication of DNA. Okay, this is something different. This is called RNA polymerase. Okay, similar name, but RNA rather than DNA. Anyway, that binds to the gene, and what it does is it moves along the gene, unwinding the helical structure of the DNA, okay, and then unzipping it, i.e. breaking the hydrogen bonds which hold the DNA bases together. Okay, and what happens is that ribonucleotides, which are nucleotides of RNA rather than nucleotides of DNA, come in one by one, okay, as you can see them here, and they complementary base pair, okay, they complementary base pair with what's known as the coding strand, and what happens is that the sugar phosphate backbone on the RNA is then cobbled together, okay, so we end up with a single-stranded molecule of what's known as RNA. Remembering that the base pairing is slightly different, okay? So we don't have the base thymine with RNA, we have the base uracil instead. So A base pairs on the DNA, base pairs with U there, okay? C and G base pair in the same way as they do in DNA. Okay, so that's something that you learned at AS, but just to remind you of that. So the RNA moves along until there's a sort of termination sequence which stops that process at the end of the gene, and we end up with a piece of RNA. Okay, so moving to the next bit, okay, actually what I'll do just before we do that, I've got a little animation which shows you that, lasts a few seconds. Okay, so here we've got a molecule of DNA, okay, with its coiled structure, okay, and what will hopefully happen is that we will see this piece of DNA being bound to by an RNA polymerase molecule. Okay, here it goes, and we'll see that it unwinds, okay, and unzips. Okay, so here's the RNA polymerase molecule, which is about to bind to a promoter site, which is a bit just before the gene, okay, which we'll talk about later. Here's the RNA polymerase, okay, it binds to the DNA, okay, it unwinds it, okay, so it's been unwound here, and then it moves along, as you see it's moving along, okay, here, exposing these bases, and in come RNA nucleotides and base pair to the DNA nucleotides on the coding strand, okay, and as we can see we're producing this long chain of RNA nucleotides which then dissociates from the DNA, and that's happening until the RNA reaches the end of the gene, a thing called the termination site, when the RNA, single-stranded RNA molecule will come away from the DNA. The RNA polymerase also dissociates from the DNA, so we end up with this strand of messenger RNA, which if you like is a template of the coding strand on the DNA. Okay, so that's transcription dealt with. Now I want to look at translation, which is the bit that we didn't really study in any detail at AS. So the starting point for translation is when this messenger RNA molecule, which we just made in transcription, diffuses out into the nucleus, out from the nucleus into the cytoplasm, and it binds to the ribosome. Now, the difficult bit to appreciate here is that there are amino acids in the cytoplasm which are attached to these special molecules here, which are called transfer RNA, okay, or tRNA for short, tRNA. Okay, so they're made of RNA, and they're cloverleaf shaped. Okay, and the key feature of these is that at one end they have a loop of RNA nucleotides which presents 3 RNA nucleotides. Okay, and that is known as an anticodon. Okay, and this is specifically 3 nucleotides there. At the other end, there is an amino acid attached. Okay, the clever thing is that these 3 nucleotides here always correspond to a particular amino acid there. Okay, so these tRNA molecules bring amino acids to the messenger RNA on the ribosome, and the complementary base pairing allows the sequence of bases on the mRNA to be read by these triplet codes, anticodons, on the transfer RNA, and this particular anticodon will always correspond to a particular amino acid. there. Okay, and again, what I've got is a nice little animation of that happening. So here we go. So what I've got here is I've got the smaller part. Ribosomes are made out of two subsections. Okay, they're made out of a small subunit, and this bit up here is a large subunit. Okay, so the RNA attaches to the small subunit. Okay, and then the large subunit attaches to the small subunit, and one of these tRNA molecules. And remember, I said that all proteins start with methionine. So the tRNA molecule bringing methionine comes and binds to AUG, complementary base pairing there. So here we have the large subunit, okay, and the tRNA molecule with its amino acid attached. Okay. Now what then happens is that another tRNA molecule comes in and binds to this part of the molecule here. You don't have to know the names of these sites here. Also binds to the ribosome, as we'll see in a second. So in comes another tRNA molecule. Okay, so this has got a different anticodon and a different amino acid associated with it. Okay, and it binds by complementary base pairs here, C to G, C to G, and G to C. Okay, then what happens is a polypeptide bond forms between the two amino acids. Okay, then what happens is the ribosome moves along the messenger RNA. The transfer RNA is released, the first transfer RNA is released. The next codon, triplet code, or codon as we call it, is then exposed, and another tRNA molecule comes in with its associated amino acid. Another peptide bond is formed. So now I've got a tripeptide. And then that tRNA molecule is released, and another tRNA molecule bearing another amino acid comes in, another peptide bond, and that process continues, keeps continuing, so the polypeptide gets longer and longer and longer, okay, until we encounter a stop codon, okay, for which there isn't an amino acid, which is coded for by this particular codon, and the whole process is interrupted. Okay, the ribosome splits apart, and the tRNA molecule goes off, and we end up with our polypeptide there. Okay. So going back to our original slide, the first 4 bullet points refer to transcription, which is happening in the nucleus, and the next is a bullet point description of what we've just seen on the little animation. So messenger RNA attaches to small unit of ribosome, tRNA molecules which have 3 base anticodon that correspond to specific amino acid bind to codon on their mRNA, the codon being a triplet code on the mRNA. The second tRNA with its corresponding amino acid comes to bind to the next codon on the messenger RNA. Peptide bond forms between the two amino acids. That requires ATP. And then ribosome moves along the messenger RNA to expose next codon, whilst first tRNA leaves. Third tRNA binds to ribosome. Process continues until stop codon encountered. Process is disrupted, and polypeptide breaks away from last tRNA molecule. Anyway, I hope that is a useful presentation on protein synthesis.